Okay, perfect.
Okay, cool.
Welcome you, and I'm so happy that you're in the webinar for Kweller Prep's AP Bio exam overview. We have an expert tutor who is now a medical student. He will tell you more about himself, but Sam is extraordinarily well educated in the sciences. He's actually so good that he taught classes, lecture halls of 300 plus students in undergraduate while at St. John's University. So Sam also has experience not only teaching privately, not only teaching to a group, but I would really love it if you could expand upon your outstanding credentials, because it's very common at Kweller Prep that the tutors are on the younger end, but the credentials are just phenomenal. And, you know, I'm all about the young talent. I love it. So I think it would be great if you could start the webinar just by giving us an overview about yourself, what you've taught and what you've prepared, and then we'll jump right in. We're waiting for one or two more anyway. So please, without further ado, it is truly an honor to know you. It's really impressive that I want everyone to know that while I was editing Sam's bio, I actually said, wow, like multiple times. You just have amazing credentials. And I totally want the whole group to hear that. So go right ahead, take over.
Thank you very much, Francis, for that warm intro. So like Francis said, I am Sam. I'm a first year med school student. I'm a first year med school student. I go to SUNY Downstate. A little bit more about me. I finished undergrad at 19, and then I finished my master's when I was 20. Like Francis mentioned, I was an adjunct professor at St. John's for about a year and a half, and I taught courses in biochem, molecular bio, cell bio, introductory biology, and yep, I think that's about it. I've been doing 3 years of molecular neuroscience research, and that was at St. John's, and now I'm in a few different labs here at Downstate. So last semester I taught AP Bio for the first time with Francis's group, and you know the results were as we expected, really good. A lot of my students got 4s. I think many people got 5s as well. So everyone got the target scores that we were looking at. Okay, so I prepared a little bit of a broad overview of what the AP Bio exam is, because I'm sure that you know some of you may be really familiar with it, some of you may be not so familiar with it. So I kind of tried to design something that I think would be really fun and useful for everybody. And let's just get started. All right, so people always ask me, Sam, you know, what is this AP exam? Do we get credit for it? Is it just something that we have to take? So what's the situation? So if you get a 3, 4, or a 5, you will get college credit. Yeah, college credit, basically free money for your AP class that you took. So if you take AP Bio and you get a 5, which many of my students do, and then you go to any legitimate undergraduate institution, they will likely transfer that and let you place out of that bio class that you took as a general rule. And this does look impressive when you're applying to your graduate schools. The fact that you stepped out of a bio class because you took an exam and you got a really good score on it in high school. That's the kind of stuff that people get very excited on when you talk about at interviews. So it's definitely in everyone's interest to do really well on the AP Bio exam. Like Francis mentioned, I did teach undergraduate level classes at St. John's. I taught some higher level bio classes, some lower level bio classes. And one thing that I noticed was that students that had a really strong biology content coming in were able to really excel at these courses at a level that students that maybe didn't get such a high level of understanding weren't able to do. So I thought that was really cool that the students that prepared well for the high school level bio, not just AP Bio, but high school bio in general, did do very well in undergrad as well. And that's something that I've seen as a professor at St. John's. Okay, so the breakdown of the AP Bio exam itself, right? So we have 3 hours for the exam. We have 60 multiple choice questions and 6 free response questions. Okay, and I'll get into each of those in future slides. When is the exam, right? So the AP Bio exam, generally speaking, it's going to be May 11. It's going to be 12 p.m. and it's going to be at whatever your local time is, which I'm assuming for most of us that's Eastern Standard. But this is a general rule. Schools have been shown to have exceptions to this, so just make sure that your school is following the norm here, and that they start that they're going to have their exam on May 11th at 12. Okay. So like I said, there's a multiple choice section, right? We have 60 questions. We have an hour and a half for those 60 questions. That gives us about a minute and a half per question. So outside of just knowing content, we have to have a really, really good strategy when we're approaching each question. I tell this to all my students. The AP Bio exam doesn't test knowledge of biology. The AP Bio exam tests knowledge of how well can you take the AP Bio exam. And you'll get exactly what that means, if you guys do decide to sign up for the full course, right? So we have individual questions where it's just going to be a standard multiple choice. Really, what you'd expect, they'll give you here's maybe a protein, here's maybe DNA, here's maybe RNA, describe, right? And you'll get 4 or 5 different descriptive options, and you just have to pick one of those options. Okay. You will get small passages, right? So you get a passage, and then next to that passage, you'll have 2, 3, maybe 4 questions that relate to that passage. So it's also really important that we develop the skills of critically reading through a short passage or a short answer section and answering the subsequent questions, because that's really key. Okay. That's the multiple choice section. The free response section, we have 6 essay format questions. All right. 2 are a little bit longer. These are, essays with paragraphs. 4 are a little bit more short answer, maybe 4, 5, 7 sentences if you're feeling really confident. I listed some of the basic topics that they cover here just as kind of a broad overview. Scientific investigation, conceptual analysis. All this is relatively vague, but we'll go through more of it when we have the actual course. Right. So like I said, there's 90 minutes for the free response section as well. And the MCQ and the free response, they're each worth 50%. Okay. Our longer questions are going to be about 8-10 points each. Okay. The shorter questions are closer to 4 points. And occasionally there is a long question that, makes you write a graph too. So it's not just going to be about writing paragraphs. It's also going to be about making sure that your handwriting is really, really pretty when you're drawing out these graphs for your graders. Okay. So now we're just going to do a little bit of a broad overview. Okay. There are 8 units that we're going to talk about. There are 8 units that are going to be on your exam. So unit 1, chemistry of life, right? What's biology? It's just chemistry. They simplified to make it into biology, right? So all of biology is just chemistry. And these are principles that when we go through the course, we'll go through in more detail, but just as kind of a broad overview. So how much is the unit 1 section, the intro to chem, or however you want to call it, how much does that weigh on your 11%. In terms of the amount, just to give you an overview of what your AP Bio class in high school is going to be like, this would take us about 5 to 7 classes to cover in a regular high school AP Bio course. Okay. Okay, over here I just have some general overviews about what's going to be a subsection under our Unit 1 Chemistry of Life exam. All right, so the structure of water and hydrogen bonding. As I'm sure some of you maybe have heard of at this point, water is what's called a polar molecule. All right, water has what are called polar bonds, and that point is probably something that each of you is going to need to know really well over the course of your next 5-10 years studying. So that's one of my favorite things. I do talk about that a lot in each of my AP Bio courses. It's a really, really important thing, not just for the AP Bio exam, but also for your futures as, whatever it may be, pre-meds, graduate students, whatever you do decide to do later on in life. Okay. Elements of life, right? So these are your periodic table elements, introduction to biological macromolecules. So we have proteins, carbs, lipids, et cetera, et cetera. Properties of the biological macromolecules. We're just going to be going over, what does a protein do? What does a lipid do? What do those words mean? Like, what do those carbon structures actually look like? And we'll go through all of this in depth in the course itself. Okay. Structure and function of biological macromolecules. That's exactly what I was saying before. What's a lipid? What's a protein? How does it work? Why does it work like that? And, this is all relatively college level material. So like I said before, knowing this is going to help you guys, not just in undergrad, maybe even in grad school, but definitely for the AP Bio exam. Okay. All right. That's unit 1. So unit 1, we talked about the chemistry, we talked about the chemical interactions, all that stuff. So unit 2, how do these chemical interactions organize into cells, right? And that's cell structure and function. The grid image here, that's the closest thing that I could find to a cell. I think it's kind of pretty, that's why I included it. All right. So this is going to be worth about 10 to 13%. Welcome, Kavita. This is going to be worth about 10 to 13% on your exam. And it's going to take you 11 to 13 class periods to cover the second unit on your actual AP Bio courses. Right? Okay. So just like we did for unit 1, for unit 2, this is all about the cell. The cell has friends inside of it called organelles. These organelles relative to eukaryotic cells are surrounded by what's known as the phospholipid bilayer, but each organelle is also surrounded by its own little bilayer. So, you can think of the cell as one big apartment building, and each organelle is its own little different apartment. And these apartments, they interact, they talk to each other. And we'll be going through how all of that happens in the course, right? So that's basically unit 2. Okay. Unit 3, cellular energetics. So outside of the chemical aspects of what goes on in a given biological cell, they also have physical aspects, right? So there are certain laws of energy that cells have to follow because cells are physical constructs and laws of energy are guided by physics. And we'll be talking about each of these. I mean, I'm sure all of you have at some level heard concepts of entropy and enthalpy. That's basically what cellular energetics are all about. Different formulas that we use for calculating really, really important things that tell us how the cell functions and why the cell functions like that. All right. Okay. That section's worth about 12 to 16%. So as you guys can see here, there's a general trend, right? 8 sections. Each section is roughly speaking weighted equally, and this will be your 14th through 17th class periods at your actual AP exam. Okay, so I kind of talked about some of these principles here. So enzyme catalysis, that talks about all those formulas that we mentioned, right? So the formulas of how the cell works, why the cell works, et cetera, et cetera. Unit 4, cell communication and cell cycle. So imagine the apartments that we talked about in the previous example. They want to share information with each other, and our cells, they have certain pathways to do this, right? So we're talking about what's known as signal transduction pathways. And it's actually really funny. I am learning about this right now in med school too, albeit at a little bit of a different level. So again, relative to anyone that wants to go to med school, grad school, whatever it may be, fundamental knowledge of this will help you long term. Very, very high yield unit, not just for your AP Bio exams, but also for your future career as biologists, medical doctors, whatever. All right, this is going to be worth about 10 to 15% of your AP Bio exam. So again, we're seeing the pattern of even distribution across all 8 units. It's going to take about 9 to 11 class periods for us to cover. Covering your AP Bio courses in your schools. And like I said, imagine these apartments are talking to each other. They have different pathways. We talked a little bit about signal transduction. The cell likes to respond to changes in stress. So the changes in signal transduction pathways are going to help the cell respond to changes in stress. Your body's based on, one general principle, which is you never want to get too high, you never want to get too low. So you have these different feedback mechanisms that have evolved to kind of keep everything at a general homeostasis. Okay. The cell cycle. I'm not sure how much exposure everyone has had through like previous bio courses, but the cell likes to grow and sometimes the cell likes to not grow. So that's the cell cycle. When does the cell grow? How does the cell grow? When does the cell not grow? That's kind of broad overview. And just like everything else, every single step in every single cell is very, 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 very highly regulated. So you will have a lot of regulation of the cell cycle, certain proteins, certain stressors. We'll go through all of that throughout the course as well. Okay. Unit 5, heredity. Shout out Harambe, but Unit 5. So heredity. This is really we're going to be going through different major principles in genetics. That's the main ideas. So there's something known as a Punnett square. That's super high yield, just kind of helping us map out genes and how certain genes work. This is worth about 10% of your exam. You have about 9 to 11 class periods to handle this. All right. And, I mentioned Punnett squares. Punnett squares are under what's known as Mendelian genetics. You guys will see 5C, we have what's known as the chi-square hypothesis testing. That's a principle in biostatistics that basically helps us understand whether something is statistically relevant or not. And we'll go through what that means in the course. But, chi-square hypothesis testing, like I'm doing this in med school right now. So again, knowing this at the level of your AP Bio exam will help a lot, okay? So we have Mendelian genetics. We also have what are known as non-Mendelian genetics. The difference is a little bit subtle. We'll go through that during the actual exam. Let's see, the cells.
Sam, can you make it a little larger so it's easier for the screen?
Sure. Is that better?
Yeah, that's perfect. Okay, great.
Awesome. Okay, so that's pretty much it for unit 5. 5.1, that's meiosis. So we talked about the cell cycle before. The cell cycle basically happens when the cell is growing. Meiosis is what happens when the cell is replicating, in a sense. All right, unit 6. So units 5 and 6 are pretty closely related. Unit 6 more talks about genetics. So on the right, there's a picture of DNA. You will need to know the structures of DNA, the structure of RNA, and how the DNA and the RNA get inherited and passed down from generation to generation. That's genetics, right? Generation is a long form of the word genetics, basically. This is worth, like we said before, somewhere around 10%, closer to 12 to 16. And we'll take your AP Bio high school exam, your AP Bio high school classes, about 18 to 20 class periods to cover. Okay, so we talked about DNA and RNA, right? So DNA and RNA, they are certain conglomerations of different macromolecules called nucleic acids. Okay, your DNA and your RNA have different structures. DNA and RNA, they get passed down to future generations via DNA replication. However, you also have DNA replication that happens to your cells, so we'll talk about that. Transcription is basically how you go from the language of DNA to the language of RNA, and translation is how you go from the language of RNA to the language of proteins. We'll talk about all of that throughout the course, but again, so this is more about DNA, RNA, patterns of mutations, what exactly mutation means. Is a little bit in the weeds right now, but we'll talk about that in the course. Just a broad overview of DNA and RNA. Okay. 7, natural selection. So natural selection, falls under Darwin's concept of evolution, right? So this idea is that mutations are random, and these mutations, whether they kind of adapt and form certain beneficial structures for a species over time, they stay. That's the main idea behind Darwinian evolution. So, we'll learn about the different principles of different types of evolution. You have natural selection, which is what I just talked about. Artificial selection is a little bit different. That's more when we consider like humans involved in this species. So, for example, think how wolves eventually got domesticated into dogs, right? So that's kind of broad concept of artificial selection, right? Okay, some nice plants here. So unit 8, fitting, this is the last unit. This is ecology. This is probably the most boring unit relative to how I like to view things, and it basically covers plants, plant responses to environment, as well as kind of zooming out from the smaller picture of the cell that we've been talking about into the kind of broader picture of population ecology, right? And this is basically where, what are the trends of different peoples, different groups, different ethnicities across different populations. So you do need a little bit of knowledge of what's known as public health stats to kind of analyze that. Community ecology, biodiversity, disruption to ecosystems. Again, these are all concepts that we're going to talk about throughout the actual course itself. But this is probably a little bit on the lower yield spectrum in terms of what you're going to need to know for the future. So it's personally not a topic that I focus on the most. And you still need to know it a good amount for AP Bio, but just keep in mind that it's not going to be as relevant as topics 1 through 7. Okay. And I think that's it. Let's double check here. Yeah. So I did want to make sure that we had a good amount of time left over for questions. So does anybody have any questions? Okay, I'm just going to check the chat, see if anybody wrote anything over the course of the
Are we
meeting.
good, Sam, or do you need anything from my ends?
I think we're good. I think we're good to go.
Okay, can we go to that college board right now? Everyone, I want to take a moment and just direct you. Sam, remember I emailed you the college board. I just want
Yes.
everyone to take a look at the college board file. So everyone, please pay attention. Sam is going to go to the college board file and he's going to, do you mind actually, can you also put the link in so that all the students have it? This is a very important file. It gives you a very clear outline of what to expect on the AP Bio. And I emailed it to Sam earlier, and he's also going to put it into the chat box so that everyone has it. And if you don't mind, oh, that's perfect. And then if you don't mind opening it, share screen. And if you could just kind of outline what they're going to see, I think that's the best way to, use our next 30 minutes wisely is to go through, so everyone, we just put this in the chat so that you'll all see it. So can you guys please, let's go through this, okay? And please scroll through it so that everyone's aligned and on the same page with what to expect from the AP course and exam description so that you know what you're getting yourself into. Okay, go ahead, Sam, you can scroll.
All right, sounds good. Thanks, Francis. Okay. So the AP Bio exam, the 2022 version is going back to how the AP Bio exam was pre-2020. I just kind of wanted to add that in.
Do you mind making it larger? I just want it to be easier for the group to see. So just hit, perfect. That's exactly what I want. So it's easier. And if you can go to the concept outline, so we'll go higher up to the concept outline. So just scroll down, 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 like page 6, 7. Yep, that's what I wanted. Perfect. Okay, good.
Perfect. Okay.
A little higher. Yep, there we go.
Okay.
Basically. Okay. All right. So we'll go through these pieces. All right. So I was actually going to go back to page, concept outline. That's exactly where we should start. Okay. Go right ahead. So if you can just, I want everyone to see this. Thank you so much for making it larger. I love it when everything is easier to see. Okay. Go ahead.
Okay. So big idea 1, right? The process of evolution drives the diversity and unity of life, right? So there are different aspects to the theory of evolution. We talked a little bit about Darwinian evolution, right? That's the second sentence here. So Darwin's theory. We do have evidence from a variety of scientific disciplines. And here, variations that occur in individuals in your population, the theory is that's how we got to where we are today, right? It's a really important scientific theory. And, as such, it is something that's going to be featured heavily on your AP Bio exams, right? So due to competition for limited resources, individuals with favorable variations in phenotypes are past traits to genes. That makes sense, right? So we didn't really know during Darwin's time, but the idea is that DNA mutations are random and that random mutations will accumulate and kind of form relative to the need of what the organism is going through. Those random mutations will form beneficial mutations, and the beneficial mutations will eventually accumulate, and that's how we see populations evolving over time. That's the main idea of Darwinian evolution. Okay, so that was the process known as natural selection, right? So in addition to that, you also have catastrophic events and events that human beings induce, right? So we talked a little bit about artificial selection, right? So imagine domesticating a wolf into a dog. That's an example of artificial selection, as well as we have these random environmental changes that can also bring us to alterations in gene pools of populations. Okay, and smaller populations are obviously more especially sensitive to these forces. The idea is that a smaller population has in it a much less diverse gene pool, and in that less diverse gene pool, you would have, let's say that a catastrophe would hit. There would be much less of a response because the idea is that the gene pool is much less developed. Okay, a more diverse gene pool or a larger gene pool, on the other hand, is vital for the survival of species because environmental conditions change. We kind of talked about that a bit. Mutations in DNA, which we talked about, and recombinations during meiosis, which remember we said is kind of how the cells have sex, so right, that's meiosis. That's meiosis is cell replication, are all sources of variation. Okay, artificial selection, aka human-directed processes, also result in new genes. Remember, think wolf going to a dog, and different combinations of different alleles that will give you a new phenotype for the same genotype. Now, what's a phenotype? So let's say that your hair is a phenotype, but the genes that encode for your hair, that's the genotype. So phenotype is like a characteristic. It's like you're giving a quality to something. Genotype explains how that quality got read from your genes. That's the idea. There are a couple of different mathematical approaches. So alleles are basically organizations of genes, copies of genes, and allele frequency can be modulated mathematically, so we can kind of predict how these alleles would change and react with certain formulas. That's the idea. All right. And this also gives evidence for the occurring of evolution in the population. Again, it's kind of changing in allele frequency. Okay, so scientific evidence supports the idea that both speciation and extinction have occurred throughout Earth's history. For example, in terms of extinction, think famously, the huge asteroid that got rid of the dinosaurs, right? That's kind of the famous extinctive event that everybody knows. Obviously, there are many more, and we'll go through that throughout the course as well. But broad picture, think the rock that knocked out dinosaurs. Okay, life continues to evolve within a changing environment. And the theory is that this would explain the diversity of life. New species would arise when two populations either diverge from a common ancestor and become reproductively isolated, shared conserved core processes in genomic analysis. Genomic analysis, basically done using different mathematical concepts, different formulaic concepts. So again, we're using math to kind of map out the genome. That's the idea. Support the idea that all organisms, archaea, bacteria, and eukarya. So eukarya, those are what we call our cells. Bacteria, that's what we call bacterial cells. And archaea are what we call a certain type of cell called archaea. A little bit lower yield, a little bit less important to know, but we'll go through exactly what that is throughout the course as well. Okay. Both extant and extinct are linked by lines of descent of common ancestry, right? So the idea is that eventually, at a certain point, archaea, bacteria, and eukarya all had lines of common ancestry. That's the idea, right? We have elements that are conserved across all three of these different domains. So bacteria have DNA, eukarya have DNA, and bacteria and eukarya also have RNA, although, what they do with this RNA and what that RNA looks like many times is like really, really different. But the idea is that, you do have DNA and RNA that are inside bacteria and are inside eukarya. That's the idea. There's universal genetic code. So that genetic code is basically how your DNA gets read. Right, so I think we briefly mentioned this before, but your DNA basically gets made into RNA, and your RNA basically gets made into proteins. Right, that's what we're saying when we mean that we're reading your genetic code for information. You have a wide variety of metabolic pathways here too. So I'm not sure. That's basically how your body explodes sugar, it's energy from it. It's a pretty cool pathway. We'll talk about that during the course, but that's an example of a metabolic pathway, how food goes from the food into your mouth to the energy that you use for your body to do things. Okay. Phylogenetic trees graphically model evolutionary history. So phylogenetic tree is basically just a bunch of lines that, try to show how one species is related to another by making lines between those species. That's really it. And the concept known as descent with modification. Okay. So some organisms and viruses can transfer genetic information horizontally. So for example, in bacteria, so let's say that one bacteria has a special gene that, is going to make that bacteria resistant to ampicillin. That bacteria can conjugate, that fancy word for talk to, a different bacteria and just spread that antibiotic resistance that the first bacteria had onto the second bacteria. And that's a really important concept that you learn about in med school too. For those of you that go to med school, this is one of the things that is really important, not just for AP Bio, but for future undergraduate and hopefully graduate level coursework as well. So that's really cool. Okay, so the theory is that the process of evolution explains the diversity and unity of life. but an explanation of the origin of life isn't clear just yet. So we do have some experimental models that support the idea that chemical and physical processes on what are called primitive Earth. So this is basically just Earth before cells, Earth before us, could have produced these, complex molecules that we see as proteins, lipids, phospholipids, whatever it may be, and really, really simple early cells. That the idea is that, all the organisms came from those early simple cells. Under lab conditions, complex polymers and self-replicating molecules can assemble spontaneously, right? So there is laboratory evidence that under very controlled laboratory conditions, we can replicate what we think primitive Earth might have looked like, and create a kind of hypothesis of what the origin of life could have been based on that, speculating based on the laboratory conditions. That's the idea. First genetic material may not have been DNA. That's a little bit more of a historical thing, not necessarily something important to know, but short sequences of self-replicating RNA that may serve as templates for polypeptide synthesis. So that's a hypothesis known as the RNA world. That's basically saying that everything was originally RNA, and because RNA is much more flexible, RNA is much more usable than DNA, so it makes much more sense that everything was originally much more flexible RNA that then became these kind of more complicated forms, primitive cells, eukarya, bacteria eventually. So that's the idea. Okay. Protosymbiotic formation was most likely followed by the evolution of different primate groups of bacteria, these various means of obtaining energy. Okay. Mutually beneficial associations among bacteria are thought to have given rise to eukaryotic cells. So the idea is, how did a eukaryotic cell get here, right? So maybe one bacteria ate another bacteria, and that second bacteria kind of enslaved, literally what happened, kind of enslaved the first bacteria, and that's how you got the organelles. Remember the organelles, that's that apartment that we talked about, the apartment that's inside the apartment building. So the idea is that one big bacteria ate a little bacteria and then enslaved it. That's the main theory as to what's known as the protosymbiotic theory in terms of how eukaryotic cells came about. All right. So change in the genetic makeup of a population over time, that's the concept that we call evolution, right? So natural selection is the major driving mechanism of evolution. The essential features of the mechanism contribute to the change in genetic makeup of the population over time. And Darwin's theory of natural selection states that the inheritable variations occur in individuals in a population. Due to competition for resources that are often limited, individuals with more favorable variations of phenotypes are more likely to survive and produce more offspring. That's the idea, thus passing traits to subsequent generations. The concept known as genetic fitness, the number of surviving offspring left to produce the next generation, is, according to the theory, a measure of evolutionary success, right? So if it works, it got kept. That's the main idea, right? So we measure success by what got kept, because what got kept, according to the theory, works. That's the main idea. Individuals themselves don't evolve, but the concept is that populations involve. So populations made up of individuals, that is what undergoes evolution. The idea is that individuals themselves, relative to Darwin's theory, don't undergo evolution as is being explained here. But the environment is always changing, and there's no quote unquote perfect genome. And a diverse gene pool is needed for survival disease. Genetic variations in the population can also contribute to the diversity of the gene pool, right, which makes sense. Variation contributes to diversity. Right? Changes in genetic information could be silent, with no real observable phenotypic effects, or they can result in a new phenotype, which can be positive, negative, or neutral relative to the organism. The interaction of the environment and the phenotype actually determines the fitness of the phenotype itself. Thus, the environment does not directly changes in DNA, but acts upon phenotypes that occur through random changes in DNA. These changes can involve alterations in DNA sequences, changes in gene combinations, and the formation of new gene combinations. Although natural selection is usually the main mechanism for evolution, genetic variation in populations can occur through different processes, including, but definitely not limited to, mutation. We talked about mutations a little bit, right? So DNA, when it replicates, it mutates. So it replicates, mutates, that's the idea. Genetic drift. So genetic drift is basically the idea that a certain population drifts, goes away from, however you want to call it, an original genetic composition due to environmental stresses, due to environmental factors. That's how you get genetic drift. Sexual selection and artificial selection. So we did talk about artificial selection. Think human domesticating a wolf into a dog. And sexual selection, for example, the idea there is that traits that were found desirable is how people got together. It's how that was the origination of sex and monogamy. A little bit of a lower yield concept relative to AP bio. Yeah. Inbreeding, so brothers and sisters having sex, small population size, non-random mating, absence of migration, and a net lack of mutations could also lead to a loss of genetic diversity. Human-directed processes such as genetic engineering can result in new genes and combinations of alleles that will confirm new phenotypes. The theory is that biological evolution, as driven by natural selection, is supported by some evidence from many major scientific disciplines, including, but definitely not limited to, geology and physical science, as well as embryology, but that's probably a little bit lower yield for the AP bio. Exam, and in addition, certain biochemical, morphological, and genetic information from existing and extinct organisms support the concept of the theory of natural selection. Phylogenetic trees serve as dynamic models that show common ancestry, while geographical distribution and the fossil record link past and present organisms. Okay, so this is basically just a summary of what we talked about before. I'm going to kind of breeze through it just so that, if you guys want to, you can go through that on your own pace. These are also just more summaries. Okay. Just wait to get to the second big idea here. More summary, more summary. Okay. So life continues to evolve within a constantly changing environment, right? So speciation and extinction have occurred throughout the Earth's history, and life continues to evolve within a changing environment. But the rates of speciation and extinction vary. Speciation can be slow and gradual, or as described by the theory known as punctuated equilibrium, which this theory basically states that there are certain gaps in the fossil record, and we explain the gaps in the fossil record by certain necessary, what's called the quote-unquote explosions of life that explain the gaps in the fossil record. And that's punctuated equilibrium. All right, so those are those bursts that we talked about, right? Think the explosions to explain the gaps in the fossil record, followed by relatively quiet periods. Certain times of ecological stress on a given environment, extinction rates can be rapid, and mass extinctions can be followed by adaptive radiation, the rapid evolution of species when new habits open. Academic or scientific evidence, including emergent diseases, chemical resistance, and genomic data, support the idea that evolution occurs for all organisms, and that evolution explains the diversity of life on the planet. That's kind of the main scientific concept of what evolution does, right? Explains diversity. That's the idea. A species can be defined as a group of individuals capable of interbreeding and exchanging genetic information to produce viable, fertile offspring. New species can arise when two populations diverge from a common ancestor and become reproductively isolated. Although speciation can occur by different processes, reproductive isolation must be maintained for a species to remain distinct. Evidence that speciation has occurred includes fossil records and genomic data. All right. So more summaries, more summaries, more summaries. The origin of living systems is a natural process. So the process of evolution can explain the diversity and unity of life. A number of different experiments to provide evidence that the conditions on early Earth did provide an environment that were actually capable of generating these different complex organic molecules that we talked about. Think, lipids, proteins, et cetera, and smaller cell-like structures as well. Think, we talked about that primitive cell. But think about that too. For example, there's a model known as organic soup, right? And basically, the idea here is that the primitive atmosphere had certain inorganic precursors like rocks and lightning, from which organic molecules could have been synthesized. So this is a bit more speculative, but the idea is that they could have been synthesized through natural chemical reactions as catalyzed by the input of energy. In turn, these molecules served as monomers, think building blocks, for the formation of more complex molecules. So basically, rock gets hit with the lightning, and the amount of energy of that rock, the theory is, maybe could have given it the kind of boost that it needed to become organic. Organic meaning carbon containing. And all these different proteins, DNA, RNA, they are also organic, and meaning that they also have carbon. That's the idea. All right. So where are we? So some models suggest that primitive life developed on biogenic surfaces such as clay, right? So the idea is that maybe clay has certain properties that make it more suitable for the development of biological life, right? That's called a biogenic surface, right? So maybe this clay kind of helped, and that's how the primitive life developed. That's the theory, right? So these biogenic surfaces, like the clays, could have served as templates and catalysts to assemble macromolecules. So again, they could have helped the more simple proteins and lipids become more complex macromolecules as well. That's the idea. Okay. Under lab conditions, complex polymers and self-replicating molecules can also spontaneously assemble, right? So that kind of adds a little bit of support to this theory on some level. It remains an open question whether the first genetic and self-replicating material was either DNA or RNA. Okay. Yeah, I'm going to skip through the summaries. You can read them on your own time. I just thought it'd be better to go through the more in-deep material with you guys, and then you read the summaries on your own time. And yes, we did send this link in the chat. Cool. So the second big idea, biological systems can utilize free energy and molecular building blocks to grow, to reproduce, and to maintain a dynamic homeostasis, right? So we did talk about how the body never wants to get too high or too low. And we have these certain mechanisms to keep us in the middle. That's the idea. Living systems need free energy and matter to maintain, order, grow, and reproduce. Organisms employ various strategies to capture, to use, to store free energy and other vital resources. Energy deficiencies are not only detrimental to individual organisms, they also can cause disruptions at the level of a population and even wider at the level of the ecosystem as a whole. Autotrophic cells capture free energy through photosynthesis and chemosynthesis, and photosynthesis traps free energy present in the sunlight that in turn is used to make carbs from CO2, carbon dioxide. Carbon meaning one carbon, dioxide meaning two oxides, hence carbon dioxide. Okay. Chemosynthesis can capture energy present in inorganic chemicals. For example, think the stuff that makes up rocks, and cellular respiration and fermentation. These are the different metabolic processes that we talked about. Glycolysis, which is basically the explosion of the sugar molecule. That on some level can be considered cellular respiration and fermentation relative to this class. And it can harvest free energy from sugars to make free energy carriers, including ATP. The idea is that the free energy available in sugars drives certain metabolic pathways, and photosynthesis and respiration are dependent on one another. Okay. Cells and organisms need to exchange matter with the environment. For example, water and nutrients are used in the synthesis of new molecules, and carbon moves from the environment to organisms where it is incorporated into carbs, proteins, nucleic acids, and fats, right? So carbohydrates, right? That term means carbon and hydrate, right? What does it actually mean? It's basically just some combination of carbons and hydrogens and sometimes other stuff like oxygen and nitrogen, but carbohydrates are basically just different units of organization, different patterns of carbon and hydrogen. Okay. Proteins. So normally you're going to have, carbon, hydrogen, oxygen, nitrogen, and sulfur. Those are your main elements that are going to form and interact with different proteins, nucleic acids as well. So remember we talked about DNA, RNA, acids. Talking about lipids, those are related to fats. And oxygen is necessary to make really, really efficient free energy use in cellular respiration. The exact mechanism here is a little bit complex, but the basic idea is that when oxygen is used in a eukaryotic cell, it makes us use the metabolic pathways that get the current of energy, known as ATP, much, much faster. And that's why oxygen can be used really, really effectively to help us get ATP. All right. Differences in surface to volume ratios affect the capacity of a given biological system to obtain resources and eliminate waste. Programmed cell death, also known as apoptosis. This is when the cell kills itself. But this is when the cell kills itself in a good way, because chances are it doesn't want the cell to progress into cancer, right? So if you don't want to progress into cancer, the cell kills itself. That's the idea. Kind of morbid, but that's the idea. Okay? So apoptosis plays a key role in normal development and differentiation. The other word for differentiation that they use here is morphogenesis. Okay. Membranes. So membranes are basically just made up of what's known as a phosphate group, which is basically just a bunch of elements organized together. and attached to a lipid, or think fat. So it's a phosphate group attached to a lipid. We'll talk about what that actually means throughout the course, but membranes can allow us to create and maintain different internal environments that differ from external environments, right? So the structure of these cell membranes that we talked about can result in what's known as selective permeability, meaning, kind of think of the cell as a bouncer, right? Some stuff gets in, but not a lot, and some stuff comes out. But the cell can kind of select, what's going to come inside and what's going to stay outside, just like a bouncer would. That's the idea. Okay. Movement of molecules across them via osmosis, diffusion, and active transport maintains a dynamic homeostasis. And eukaryotes, which is us, internal membrane partitions the cell into specialized regions that allow cell processes to operate with optimal efficiency. Each compartment or membrane-bound organelle is actually going to enable localization of certain given chemical reactions, right? So the idea is you have a cell. Inside a cell, you have organelles, and inside organelles, you have certain chemical reactions going on, and these chemical reactions going on happen best within that specific organelle. So again, this lets us have a very, very, very specific structure of organization within the cell, right? Organisms have feedback mechanisms. So we talked about, you never want to go too high, but you never want to go too low, right? So organisms have a kind of mechanism for keeping you at a certain given dynamic homeostasis. That's how you stay nice and in the middle, by allowing them to respond to changes in their internal and external environments. Negative feedback loops can maintain optimal internal environments, and positive feedback loops can amplify responses. A really good example of this is pregnancy. Right? So in pregnancy, you have a really famous example of a positive feedback mechanism. Right? Changes in a biological system's environment, particularly the availability of resources, would influence responses and activities. And organisms use various means to obtain nutrients and get rid of wastes. Homeostatic mechanisms across different phyla reflect both the continuity due to the common ancestry of the phyla and the change due to evolution and natural selection, especially in plants and animals. Defense mechanisms. So the idea is that you have an immune defense systems, right? A parasite or an enemy comes and it tries to attack your cell, and we have certain immune or certain defense mechanisms that kind of sent out attackers against the parasite to kind of get rid of the parasite. That's the idea. Right? Additionally, the timing and coordination of developmental, physiological, and behavioral events are regulated. Higher fitness of individuals and long-term survival of populations are also regulated, right? And that's the idea, okay? Growth, reproduction, and maintenance of organization of certain living systems does need what's called free energy and matter. So free energy, think ATP. Exactly what ATP is in terms of the chemical structure we'll go through during the course, but just kind of trust me that there's this molecule called ATP. That's your body's currency for energy and matter. All right. Living systems need energy to maintain, order, grow, and reproduce. It's kind of self-explanatory. In accordance with the laws of thermodynamics to offset entropy, we talked about entropy a little bit before. You can just think of entropy as randomness at our level. Energy input must exceed energy loss from and used by an organism to maintain order. Organisms themselves use various energy-related strategies to survive, and the strategies include different metabolic rates, physiological changes, and variations in reproductive and offspring rates and strategies. Not only can energy deficiencies be detrimental to individual organisms, but also affect population size and cause disruptions at the ecosystem level. All right. Several means to capture, use, and store free energy have evolved in organisms. Cells can capture free energy through photosynthesis and chemosynthesis. Autotrophs, which are basically cells that can feed themselves automatically, hence the term autotrophs, they capture free energy from the environment, thus feeding themselves automatically, and they include energy present in sunlight, for examples, and in certain chemical sources. Famous example of autotrophs are most plants. Right? Heterotrophs can harvest free energy from the carbon compounds produced by other organisms, and through a series of coordinated reaction pathways, photosynthesis will trap the free energy in sunlight and use that to produce carbs, CO2, and water. Exactly how we'll go through during the class. Cellular respiration and fermentation use free energy available from sugars and from interconnected many-step pathways. We talked about glycolysis, which is basically just glucose exploding. We talked about the Krebs cycle and the electron transport chain. We didn't talk about the Krebs cycle and the electron transport chain. Remember, I mentioned that there are certain pathways that are more efficient to use when you have oxygen. That's where the electron transport chain comes in. That allows us to use oxygen more efficiently and make more energy, think ATP, relative to that original oxygen that we had, right? And this is all done to phosphorylate ADP, thus making ATP, right? The free energy available in sugars can also be used to drive metabolic pathways vital to cell processes. The processes of photosynthesis and cellular respiration are, however, interdependent in their reactants and products. Organisms must exchange matter with the environment to grow, reproduce, and maintain organization. However, the cell surface to volume ratio itself can also affect the biological system's ability to obtain resources and eliminate waste products. All right. Water and nutrients are essential for building new molecules, and carbon dioxide can move from the environment to photosynthetic organisms where it can be metabolized and kind of made into carbs, proteins, nucleic acids, or lipids. And this should make sense when we understand that what's carbon dioxide? It's carbon with 2 oxygens. And what's the main element? Carbon is an element. What's the main element that we're going to have in carbs, proteins, nucleic acids, and lipids? That's really going to be carbon, right? That's the main player. Hence the term carbohydrate, and hence the term carbon dioxide. That's the idea. Okay. Nitrogen is also a really key player here. It can help us build nucleic acids and proteins. Phosphorus is incorporated into nucleic acids, phospholipids, ATP, and ADP. In aerobic organisms, however, oxygen can serve as an electron acceptor in energy transformations. All right. More summaries here.
And then I just want you to be mindful, Sam, if you can
Sure.
give some closing remarks, because we're hitting the hour. So
Sure.
I think that would be great if you could really give them some closing remarks, and
Okay.
hopefully everyone enjoyed the webinar, and just give us some ideas. And everyone, please type thank you in the chat. We're going to wind down. So Sam, let's do closing remarks and
Okay.
your very last set of tips.
Sounds good. Okay. So closing remarks, I would say, like I said, the AP Bio exam does not test your ability to know biology. It tests your ability to know the information measured on the AP Bio exam. A lot of what we just talked about might be a little bit more depth than what you need. So if you're a little bit freaked out, don't worry, that's normal. Like we said, we have an hour and a half for the multiple choice section, hour and a half for the pre-response section, 3 hours for the whole thing. So, we do need to work on timing strategy. That's really where we're going to get most of the points on the exam. So, I'm sure you're all going to get 3s, 4s, mostly 4s and 5s when we see you for the full exam ASAP. Okay. And I'm just going to
someone asking, can you just give some tips for the free response questions?
Sure. If you're going to write something to a reasonable extent, make sure that it's true, right? Because I would say oftentimes students think, oh, let me elaborate, and the elaboration is itself wrong, and that can lead to your examiner being like, okay, this part is wrong. What else did the student not know, right? So now when they're grading the rest of your short answer, even though they really shouldn't do this, but in actuality they do, they might get a little bit of bias based on the fact that you said a wrong sentence when they're reading the next sentence. Even if the next sentence is correct, they're now going to be like, well, does this student really know? You really want to minimize to the max extent possible, your examiner going through that. So keep the answers short but long enough, and generally speaking, try not to say things that are wrong, because the exam that it's a very subjective grading process, unfortunately. So your examiner could get a little bit quote unquote ticked off. So trying to make sure that doesn't happen by not putting anything that you don't know to a reasonable extent is true. Okay. Check the chat. Any other questions?
No, I think this was great, everyone. I just want to say thank you for everyone for participating and for listening. And Sam, thank you and good luck with medical school. You want to give a minute of your medical school advice? What's the best recommendation you can give for those considering the application route to medical school post-college?
Sure. I would say, try to tell a story on your application, right? 3,000 hours of research doesn't look better than 500 hours of research if your 500 hours of research have a better story. So, give a story, right? Tell them who you are, tell them about you, and craft a narrative that you think that the interviewer or your medical school is going to enjoy. And obviously, really high MCAT, really high GPA. That's the priority, but try to tell a little bit of a story with your application as you go through and think, what extracurriculars did you take? Make it a story. I would say that's my advice.
That's so great. I wish everyone a lot of good luck on behalf of the tutoring community of Kweller Prep. We're going to conclude our webinar, and I want to remind you we have other webinars coming up. There's just so much knowledge about college coming up. I also just want to let everyone know Sam is available as a private one-on-one tutor. He's a platinum level tutor. He's actually been tutoring for quite some time. So we have a lot of high level talent at Kweller, and that includes Sam, and it's very impressive. And I'm oh my goodness, I definitely I don't know if I would be lecturing in front of 300 students. That's a lot. So I commend you for taking on such gigantic responsibilities so young, and good luck with medical school. We're really excited that you're at part of Kweller Prep and a part of the community and able to tutor and work with students, and give back and pass the torch. And thank you. And just to let everyone know, Sam not only tutors in the sciences, he helps with the medical school application process. He's worked with a number of students, including my own family member, who is now finishing medical school. We are going to her medical school graduation party on Memorial Day weekend, guys. So Frances Kweller will be out of town. All right. Thank you. Thank you so much. Have a wonderful day. Bye, everyone.